Everyone, my name is Grace McNamara, and today I'm going to talk to you about immunohistochemistry, or IHC, what it is, how it works, and how it's used in research, especially in studying the brain. So let's start with the basics. Immunohistochemistry, or IHC, is a tool that helps scientists find out where a protein is in a tissue, for example, your brain, muscles, or even plants. It doesn't just tell scientists that the protein is there, it shows them exactly where it is, like putting a bright spotlight on it. We can see on the right how the spotlight has been put onto these specific tissues in the colors of red, green, and blue. This helps scientists learn what the protein does and how it might affect the body. So let's break down what the word immunohistochemistry actually means. Immuno means related to the immune system, which protects your body, and specifically in this case, antibodies. Histo means tissue, like skin or brain tissue. And chemistry means studying substances and how they behave, as well as their shape and structure. So essentially, immunohistochemistry, while being a big word, just means studying tissues using special immune system tools. Here's how IHC works. Scientists use something called antibodies. Antibodies are proteins your body makes to find and stick to harmful invaders like viruses. In IHC, scientists use antibodies to stick to the protein they want to study. The first or primary antibody, like we can see on the right, sticks to the protein, or in this case, denoted as an antigen. The second antibody, or secondary antibody, sticks to the primary one. This secondary antibody has a special glowing tag, so when scientists look under a microscope, like we saw in the last slide, they can see exactly where the protein is. So in the Anton lab, or the lab that I decided to look at, which is actually a lab that I work in, they study how connections in the brain work and look. As we can see in the photo on the right, this is a mouse brain that IHC has been used on to specifically target certain proteins and have them light up so we can see where they are in the brain. Using IHC, they can look very closely at these parts of the brain and actually zoom in on them, which we can't see specifically in this photo. This helps these scientists, including me, understand how changes in the brain and their connections might affect how we think, learn, and move. Here's my bibliography for the images. Thank you so much for listening.